Welcome to the Johnson & Johnson sponsored Skin Barrier Symposium, Beyond the Skin Barrier. We're going to look at how the skin barrier is developing and what it means in the future. It's come a very long way from the pioneering works of Professor Marks many years ago. I'd ask you to switch your mobile phones off. We'd be most grateful if you could fill the evaluation forms in at the end. And these are our disclosures. The skin barrier is central to normal, perfect skin. And it protects the baby as they get older, to the moody teenager, to maturity. But the skin barrier can break down, as in atopic dermatitis. And the brick wall crumbles, that is the skin barrier, as a result of gene-environment interactions, where environmental factors are pivotal in this breakdown of the skin barrier. And then allergens can penetrate through, triggering the development of eczema. And I will cover this in the first lecture. In the second lecture, Dr. Kimberly Capone will discuss the microbiome. And these beautiful pictures from Sonia Bowell look at the microbiome. These are the normal bacteria that cover our skin, and they protect our skin, and they influence the immune system to defend us against pathogens. And these amazing pictures from Sonia Bowell, which has actually produced a microbiome picture of the whole body. And then finally, Professor Joseph Fowler will move on to the clinical relevance, onto formulation. Formulation is not just a science, it is an art. Given set ingredients, a brilliant formulator can produce something of amazing efficacy, while someone who is not good at formulation will produce something totally different. So I'd like to start with the skin barrier, how it's formed, disruption, and how we can repair it. And I'd like to start with the most important part, the team. This is our skin barrier team, headed by Simon Danby. We work with engineering colleagues, Tina Lavender's group, all of our colleagues in Johnson & Johnson, and we'd like to welcome Sonia Bowell to our slide art and design team. So this baby will not develop atopic dermatitis. They have not got the genetic predisposition. And it's a good place to start to look at their skin barrier. Why are they protected? So if we look up at the brick wall, the corneocytes, they're surrounded by the lipid lamellae and they're full of natural moisturizing factor. Natural moisturizing factor is formed by the breakdown of filaggrin. It is full of acids like pyrrolidone carboxylic acids. And these acids keep the pH of normal skin around 5. This low pH is pivotal to a normal skin barrier. The cornea desmosomes lock the corneocytes together. They need to be broken down to allow desquamation to take place, and proteases break them down. But it's very important that they're not breaking them down low down in the stratum corneum, just at the surface. So they're held in check by protease inhibitors. But perhaps even more important is low pH, because low pH switches off proteases and means that the cornea desmosomes are only broken down at the surface of normal, perfect skin. And furthermore, low pH switches on the lipid processing enzymes and helps us form the lipid lamellae. So filaggrin, breaking down to natural moisturizing factors, acids, low pH, switches off proteases and switches on the production of the lipid lamellae, the mortar, in normal, perfect skin. And then in atopic dermatitis, it all goes wrong. Beautiful work from Alan Irvine and McLean's group identified changes in the filaggrin gene, which meant there were less filaggrin. Less filaggrin means less natural moisturising factor in atopic dermatitis, therefore less acids, so the pH of the skin goes up to 7 and beyond. This high pH switches on proteases, breaking down the cornea desmosomes all the way through the skin barrier. This high pH switches off the lipid processing enzymes, breaking up the lipid lamellae, the mortar. So now allergens can penetrate through the skin. So we start with low filaggrin, low natural moisturising factor, low acids, high pH, switches on the proteases, breaking the barrier down, and switches off the lipid processing enzymes. And all of this is without environmental factors. So in atopic dermatitis, we have a barrier defect, we have low natural moisturising factor, abnormal lipid lamellae. They're very susceptible to harsh surfactants like sodium lauryl sulfate. Increased pH, an abnormal microbiome that Kim Capone will talk about, an increased susceptibility to pathogenic bacteria and viruses. The environment is pivotal to the development of atopic dermatitis. If we can change the environment and make it a positive environment, we may be able to prevent and enhance our treatment of atopic dermatitis. And then back to the microbiome, these beautiful pictures from Sonia Bauer. The body is covered in these protective bacteria. They protect by inhibiting other pathogenic bacteria. And they interact. So the microbiome affects the immune response, affects the skin barrier, and this virtuous circle, nurturing the skin barrier and the immune system. And we can change the microbiome. A single kiss will transfer 80 million lovely bacteria. And it's not just about the bacteria. Note the nose, smell, touch, and the beautiful environment. This will lead on to other things that I'll cover in my next slides. I hope this was the slide you were expecting. So sensory neural is another great aspect of the skin barrier. Smell receptors are even expressed in the skin. And our brain connects with our skin. And all of this feeds in to nurturing the skin barrier and the microbiome and the immune response. So to go now to some simplicity, here we have low NMF as a result of a filaggrin mutation in atopic dermatitis, gives us increased pH, increased protease activity, and this is a protease receptor, we get increased signaling. So if we bring in the genes and the environment, here is the genes, filaggrin, less natural moisturizing factor, less acids, high pH, high protease, high signaling through this receptor, protease receptor. Bring in the environment. Harsh soap and detergents raise the pH of the skin further, so they add to the genetic factors. They further raise protease. They further raise signaling through PAR2. So the environment interaction with the genes is pivotal to the development of atopic dermatitis. And other factors such as hard water interact with soap and detergents, breaking the barrier down. And then this feeds into high interleukin-4, which actually downregulates filaggrin. So the inflammation will decrease filaggrin and make the barrier worse. So it's a negative cycle. So the skin barrier defect as a result of gene-environment interaction is the first step in the development of atopic dermatitis. And it's modifying these environmental factors that is pivotal to prevention and treatment of atopic dermatitis. So how do we assess how they affect the skin barrier? Well, clinical scoring is very insensitive if we want to look at different formulations. Here's the very sensitive techniques. We can measure protease. We can measure the microbiome, antimicrobial peptides, natural moisturising factor, or use lasers to look through the skin. And we use these techniques to develop formulations, whether they're wash products or emollients, to work out the best formulations to repair the skin barrier in atopic dermatitis. And this is beautiful, the effect of pH. Here we have normal skin pH 5. Green is protease, red is protease switched off. And you can see pH 5, the protease is switched off. pH 7, it's switched on without any topical products being added. So the pH is pivotal to protease activity. And here we can see the effect of soap, detergents, hard water, raise pH, raise protease activity, raise PAR2 signalling. And then we can combine the two together. So here we have skin pH for healthy skin, AD without a filagra mutation, atopic dermatitis with a filagra mutation, acute atopic dermatitis. Then what we're going to do is wash the skin for 2 minutes with a harsh detergent soap. And you'll see the effect on pH with this single wash with a harsh soap is bigger than the pH increase of a filagra mutation compared to a healthy control, illustrating how important the environment is in the development of atopic dermatitis. If we want to prevent and treat atopic dermatitis, we need to change the environment. And that's what we're trying to do here. We're trying to break this progression and produce a positive environment so we maintain or raise natural moisturising factor to bring pH down to 5.5, to switch off protease and to stop protease signalling. So the first place we can start with a positive environment is optimal wash products. So we bring in our optimal wash products to lower pH, have no harsh surfactants, and so to lower protease activity. And here's an example of a comparison of different wash products. Here we have a very harsh soap, extremely harsh soap, sodium lauryl sulfate, very damaging to the skin. And we have two optimally formulated cleansers, an adult one and a baby one. And we wash these several times a day and then measure what they do to the skin barrier. And Kim Capone will show you what they do to the skin microbiome. So the first one is the effect on skin pH. And you can see the harsh soap raises the pH by about 0.5 units, while the mild wash products, there's either minimal or no raise at all. 0.5 raise in pH. How clinical significant is that? Here's the effect on transepidermal water loss. So the harsh soap raises the transepidermal water loss substantially, while there's no raise with these very mild cleansers, and the same at 4 weeks. This is clinically significant. And the effect on protease activity is substantial. So the harsh soap causes a substantial rise in protease, breaking down the skin barrier with negligible changes with these very mild cleansers. So different formulations of cleansers have a totally different effect on the skin barrier. The harsh soap is very, very destructive, breaking the barrier down, while the mild products nurture the skin barrier and keep it intact. And then water. What is water? Water has a long history in atopic dermatitis, from Howell-Williams group back in the 90s, showing a strong association of a higher prevalence of atopic dermatitis with hard water areas. Carsten Floor yesterday presented excellent data on a large cohort showing exactly the same. Strong association, increased prevalence of atopic dermatitis in hard water areas. Yet, a trial of a water softener, a conventional water softener from Howell-Williams group, showed no improvement in atopic dermatitis, no reduction in prevalence. We teamed up with Howell-Williams group in this systematic review led by Simon Danby, and the water authorities and found out that conventional water softeners actually raise the pH of the water. And that may be why a conventional water softener does not lower the prevalence of atopic dermatitis. And we're now working with Carsten Floor and Harvey Bowden and colleagues looking at how we can change water from hard to soft to improve its effects on the skin barrier and how it interacts with wash products. And that brings us finally on to optimal emollient formulations. Emollients are certainly not the same. They differ enormously in their effects on the skin barrier. So here we can see we're trying to bring an optimal emollient in to raise levels of natural moisturizing factor, to lower the pH, to switch off protease activity. And here we see two different types of emollients. Here we have a humectant emollient that contains different humectants, and here is a non-humectant simple emollient, just based on petrolatum. This emollient does not contain pyrrolidone carboxylic acid, but it induces its expression in the skin. So it's actually changing the skin's own production of this ingredient of natural moisturizing factor, while the non-humectant emollient does not have this effect. And here's another example, effects on skin pH. This is based on a study from 1953, looking at an oat-based formulation compared to aqueous cream and simple emollients. And here you can see the effect on pH, which is exactly the same as what was seen in this original study in 1953. An oat-based formulation lowers the pH of the skin, while aqueous cream and other simple emollients raise the pH of the skin. So these formulations are totally different in their effects on the skin barrier, and we would predict would have completely different clinical effects on the skin barrier, which Joe Fowler will cover later. So all emollient formulations are not the same. They differ enormously depending on how they're formulated and what they contain. So in conclusion, atopic dermatitis arises as a result of a complex gene-environment interaction, raising the pH of the skin as a result of low natural moisturizing factor, enhancing protease activity and breaking down the skin barrier. The wash products we use, harsh wash products, contribute even more than a filaggrin mutation in terms of their effect on skin pH after washing just once. And so, if we want to prevent and treat atopic dermatitis, we need to use optimal wash products, in the future optimal water, and optimal emollient formulations. Thank you very much for your attention. I'd now like to move on to Kim Capone and the microbiome. Dr. Capone is head of microbiome research at Johnson & Johnson at Skillman Research Laboratories in the US. She's pioneered amazing work looking at the microbiome in normal skin and diseased skin and the effects of topical products on the microbiome and how this should influence how we develop products in the future. So there's the microbiome hand to pass over to you. Thank you.
Thank you, Mike. Good afternoon or good morning, everyone. So today I'm going to talk to you about the evolving concepts for the skin barrier and a role for the microbiome. And as you all probably know, this is a very new and emerging field. And so we're starting to learn more and more about the role of the microbiome in skin health. And I hope to introduce you to some of the current knowledge and some of the work we've been doing and where we're going in the future. So the role for the microbiome in skin health. So we all, I think, understand the hygiene hypothesis and the idea that over-sanitization and antibiotic use may explain the increased rate of allergies and atopy in residents of industrialized countries. And so there's also a possibility that hygiene-associated changes in the skin microflora may underlie some of the diseases that we encounter, like atopic dermatitis, particularly in genetically predisposed individuals. And what's important to understand is that the development of atopic dermatitis on the skin and in a barrier dysfunction sort of way actually can lead to the development of atopy like food allergies, asthma, and allergic rhinitis. And so maintaining the proper skin barrier function is really important in preventing these sequelae. So the skin microbiome plays a number of roles, at least as much as we know today. And so the first part is healthy colonization, and that's likely obtained through immune tolerance. But it's also very important that the skin pH is physiologically relevant at that time. So around 5.5 to 6, which is normal, these microorganisms prefer that pH. So when the pH range goes outside of that, there starts to become issues with the colonization and the distribution of flora on the skin. And I'll talk a little bit about that in the context of atopic dermatitis today. Many members of your microbiome actually produce their own antibacterials that directly inhibit their competitors, and some of which might be pathogens for us. For example, Staphylococcus epidermidis produces bacteriocins that are antimicrobial against Staphylococcus aureus, which we all know is a contributor to atopic dermatitis, as well as wound infections. And the microbiome itself has been shown in recent years to actually modulate the skin's immune response. It was long thought that the gut microbiome definitely plays a role in the function of our health, but that the gut microbiome may be actually doing more of the work. And what we're learning is that indeed the site-specific microbiomes on the skin are very much influencing the immune function of the skin in that region. So it's important to maintain the proper environment, to maintain the proper microbiome community so that you have healthy skin. And so the particular organism that I was talking about that increases the skin immune response is Staphylococcus epidermidis. And I'll show you some interesting work in the next few slides that shows how that really is playing out in skin infections. It's also important to note that the skin microbiome, when you look at the entire human body, there are microbiomes in all of the various regions of our body, and the skin microbiome variability is relatively high. And that has a lot to do with the encounters that we have in the environment with our skin. So we're constantly picking up transient microorganisms, even kissing, but even touching people, we're transmitting bacteria and fungi and other things back and forth. And so maintaining the proper physiology of one's skin can help to prevent colonization of pathogens that one might pick up in the environment. But it also challenges us in this field to look at the microbiome and changes in the microbiome clinically because of this variability. So this is the study I was just talking about earlier. So it's been shown recently in 2012 by this group, it was published in the journal Science, that colonization of germ-free mice with Staph epidermidis, which is a commensal microorganism of our skin, during a Leishmania major infection, which is a parasitic infection, actually rescued protective immunity. So if you look at these slides here, this panel here, what you can see is in a specific pathogen-free mouse, meaning basically a mouse that has the proper microbiome, doesn't have a pathogen, and they're infected with Leishmania major, you can see clearly that the lesion is a large lesion. You can also see that indicated here by lesion size. When they looked at germ-free mice and they infected them with Leishmania, you see the lesion much smaller. And when you added back just Staphylococcus epidermidis, that response was rescued in terms of its immunity. So the idea here is that there's a very fine balance between protecting the skin and causing infection, and our microbiome members are providing these functions. So they're both protective, but they're also contributing to pathology. So again, maintaining this fine balance and creating an environment where they can do what they need to be doing in the proper pH and the proper environment is very important. So we're learning more and more every day about how the microbiome actually functions. And so particularly looking at the skin, we now see diagrams, for example, that show the skin cross-section as we have seen it for many years, but now also incorporating the microorganisms as well as the mites that you can see here that are living on our skin. And so there's been some work that's shown that it may be possible that these organisms are not just present on the surface, but they're actually coming from deeper in the skin. And so it's still quite controversial, but it's something to think about. Where are these organisms that colonize the surface of our skin coming from? So the idea, though, that these organisms are contributing significantly to our immune response is such that they may be helping us prevent irritation from irritants that we're exposed to, like dirt or chemicals, especially ultraviolet light. For example, it's well known in a number of papers from a couple of years ago that when people who spend a lot of time in the sun start to develop organisms in their microbiome in more number that actually provide ultraviolet benefits. So they're actually helping us maintain homeostasis there. So what I want to show here is actually the idea that infant skin differs from adult skin, and it continues to evolve over the course of life. So the idea here is that babies are not mini adults, and their skin is very different in structure and function. And it's important to keep this in context when we talk about the microbiome, I think. So what you can see here in this video microscopy or this confocal laser microscopy of infant skin on the top panel and adult skin on the bottom is that infant skin is, first of all, it has deeper glyphs in the skin, so the topography of the skin is different, which may change the way that microorganisms colonize the skin. Although not depicted here, their corneocytes are also smaller than adults, which may be an important factor as well. In addition, in terms of the function of the skin, through a measure of conductance, which actually tells you how much water is being held in the skin, it's clear that infants have more water holding in their skin than adults. But their transepidermal water loss is higher than adults. So while they have more water in their skin, their barrier function also allows more water to escape the skin. So their skin is different than in adults, and that may be important when we start to understand the function of both the skin and the microbiome in concert. What I'm showing you here in this slide is work done by Dr. Maria Dominguez-Bello at NYU a couple of years ago in 2010. And this is a really seminal paper because it's showing you the foundational microbiome of babies when they're born through vaginal delivery or cesarean section. So for example here, just for reference, this is what the oral microbiome of the mother looked like at the time of delivery. And this is mom's vaginal microbiome profile. And her baby, so this is about 5 or 6 women on average that were done in the study. And so the babies who were born by vaginal delivery clearly are exposed to the organisms in the vaginal canal. And so when you swab their skin, you can see vaginal organisms there. In contrast to that, if you look at the skin profile of the mothers before they gave birth versus the skin of babies who are born by cesarean section, you can see very clearly because they do not go through that vaginal environment, they're only being colonized with organisms that they're encountering from the people handling them, the hospitals, the environment that they're in. And in this study, actually, Gloria demonstrated that a lot of the staphylococci that these children were picking up on their skin were actually hospital isolates, which were antibiotic resistant. And so it's important to think about, is this foundational colonization, or at least inoculation, important for the future development of a healthy microbiome? And so that's really where we started looking into the microbiome beyond right at birth, beyond day one, if you will. And so we wanted to understand, because at the time the NIH was looking at, the Human Microbiome Project was ongoing, and the NIH was looking at how the microbiome on the skin evolves on adults, so we were very interested to see this in infants. And so we conducted a study in 2010, which eventually was published in the Journal of Investigative Dermatology, on how the microbiome evolves over the first year of life. So we had 35 infants in this study. About 30% of them were between 1 and 3 months of age, 4 to 6 months of age, and 7 to 12 months of age. And so we looked at how their microbiomes changed over the course of that time. But for simplicity here, I'm showing you simply what the infant microbiome looked like on average compared to an adult. And what I can tell you, and you can see very clearly, is that the distribution is different. Babies and adults have the same skin microflora present on their skin. They're just present in different distribution. And there may be taxa missing here that they eventually pick up later. But the idea here is that because babies' skin structure and function is different, so for example, very importantly, they don't produce very much sebum, they have mostly staphylococci and streptococci colonizing their skin. So almost 51% of their microbiome is these Firmicutes, which is the class that staph and strep belong to. And that's in contrast to what happens in an adult profile, where you see most, 51% of the microbiome is actinobacteria, which is driven largely by Propionibacterium acnes. And that has everything to do with the fact that adults produce more sebum, and these are sebum-loving bacteria, and so it changes the distribution. And then the feedback of these distribution changes would be very interesting to understand, and that's where active research is going into today. So to look at the profile of the microbiome in a little bit more granularity, what I'm showing here in this first panel is the same data from the pie chart and the the slide previous. But here we're looking at, on average, all the genus that you can, genera rather, that you can see distributed through all of the infants through the first year of life. We ended up following these infants longitudinally. So this year they're 7 years old, and we followed them when they were 3 and 4 years old and 5 and 6 years old. So these are the same infants, and so you can really look longitudinally at how the microbiome is evolving. And this is the distribution of their mother's microbiome. So what you can see essentially is that the amount of streptococci and staphylococci decrease as the child is getting older, and the amount of propionibacteria, which is depicted here in green, is increasing as compared to what we saw in infants. So as their skin is evolving, their microbiome is evolving with it. And again, the feedback that then these changes in the microbial distribution provide the skin may be very important in the development of healthy skin. So this also here on this right side shows the relative diversity of the microbiome. So diversity is very key when we think about the microbial communities in our bodies. So for example, in atopic dermatitis, one of the major problems there is the monocolonization of atopic lesions with Staphylococcus aureus. And so increasing diversity and maintaining diversity in one's microbiome, particularly in situations where the microflora is contributing to breakdown of the barrier, is very important. So understanding the baseline of a healthy state is where we started to look at how the relative diversity of infant skin microbiome. So basically, how many of these all of these genus, various genera are present in these samples. And so what we're observing is as these children get older, and it's very clear if you look at the forehead sample, as these children get older, you can see a slight increase in the diversity of their microbiome. And that has everything to do with the fact that the overtaking of staph and strep is reducing. So you start to see an increase in the less dominant members of the microbiome as the child evolves. And so now talking specifically about atopic dermatitis, this is work done by the NIH. So Heidi Kong and her group was looking at how the microbiome changes with treatment in atopic dermatitis in children. And so in this study, what they did was they had children who had atopic dermatitis but were currently not experiencing a flare and compared them to control patients. So here I'm showing just the distribution of the staphylococci present on their skin. So all of these, the gray part that is masked out is the rest of the profile of the microbiome that you can see for yourself in the paper if you're interested. But the point here that we're trying to illustrate is that there are less staphylococci colonizing the skin of healthy controls versus the colonizing the skin of atopic patients, even when they're not experiencing a flare. So they're carrying more staphylococci in their skin. And that may be because staphylococci have a much larger range of pH that they can tolerate, and it's known that atopic skin has a slightly elevated pH even when they're not experiencing a lesion. And so it may be that they prefer that environment and they can overtake and compete with the rest of the microbiome that I'm not depicting here. Now, when you experience an atopic lesion, what you observe is a significant increase in the colonization of that site by Staphylococcus aureus, but in addition, you see other staphylococci and Staphylococcus epidermidis also contributing to this increased colonization. So it's not known whether or not the epidermidis, as I described earlier, are part of our healthy commensals. It's not understood whether or not they are helping to try to compete with the Staph aureus here, or if they're actually contributing to the etiology of the breakdown of the barrier. And so that's some research that needs more work to go into. But when you give treatment to these patients, so be that a bleach bath, corticosteroid therapy, barrier therapy, or even antibiotic therapy, what you see is a decrease in the amount of staphylococci in those lesional areas, and the increase in diversity, which is not depicted here in these gray bars. And then post-flare, they go back to basically what they look like at baseline. But they're never exactly like a healthy control. So they're always in a state where they're more likely to develop these atopic lesions. And these staphylococci may be contributing to that. It was very interesting in this paper, Heidi and colleagues actually mentioned that they started to observe an increase in diversity of the microbiome before they saw any clinical response. So that starts to make us wonder whether or not the change in diversity of the microbiome may actually facilitate healing of these lesions faster than just using a barrier therapy alone. Obviously, also paying attention to the environment that they're in, so the pH and the lipid profile of the skin is going to be very important. So as Mike mentioned earlier, the cleansing effects that we provide to the skin is very important for us to understand how a harsh Castile soap or a mild adult cleanser or a mild baby cleanser affect the skin. So here we're looking at healthy adult women to just understand how various types of formulations affected both the skin structure and function, but also the microbiome. And this was really the first time anyone had looked at the microbiome and challenged it with topical cleansers like this to observe changes. So what we did essentially is we gave these women either the Castile soap, these two mild cleansers, and we had them use it at least once a day for 4 weeks. And we took their microbiome measurement at baseline and then at 4 weeks afterward, and so I'll show you some of that data in a moment. First, I wanted to reiterate the message that Dr. Cork provided earlier, which is that the use of this Castile soap significantly elevated the pH of the skin. And so while we look at, all of these changes are statistically significant. And so the idea here is that this is a small step pH change, but it's after 4 weeks of use and it seems to have kind of compounded. So if you look at a harsh cleanser immediately after using a harsh cleanser, you'll see a very significant pH increase. But your body then normalizes and it goes back to relatively where it was. But after 4 weeks of doing these relative perturbations, you do see a collective increase in the pH of the skin, and that's important to note. In addition, because you see that increase in pH, and because of the effects of a harsh Castile soap on the skin barrier, do also start to see more transepidermal water loss. So the barrier function is actually being compromised by this particular cleanser as well. And not, as Mike said, not as much in the other cleansers. So the effects on protease activity is also quite pronounced. And so when you use a harsh cleanser that is not appropriately pH, so the pH of this cleanser is about 9, and so when you use this on the skin where the pH should be around 5.5, it definitely changes the pH to some degree, as well as increasing the protease activity, which is significant. And so, as Dr. Cork mentioned, it's important to understand these very fine measurements and how they are affecting the skin versus what you can see when you look at just the clinical outcome, because it's not always as clear. And so the microbiome is another way to look at that. And so here we're looking at the cleansing effects of those cleansers on the microbiome communities of those women. So what you can see, and you can't see it here because I blocked it out so that it could be more visible for you, but they all started in the same place. So everyone's microbiome profile basically started in the same place. And this is what we call a principal coordinates analysis plot. And what it's showing you in these ellipses is sort of where the different cleansers are clustering. So it's basically the average of everybody's microbiome. And each of these little dots or squares or triangles represents an entire microbiome community. So it's all the samples in the study of the women that we looked at. And so after 4 weeks of use, everyone started over here and basically on top of each other. They all converged over to this area of the plot. You can see an increase in staphylococci here, and that's because we restricted the use of moisturizers in this study. So it was important to note that we saw a significant increase in staphylococci. It's also important to see here that the mild cleansers clustered on top of each other. So when you use any type of hygiene product, you're going to change the microbiome to some degree because you're influencing the system there. But the idea is that they changed in a very different way than what the harsh cleanser did. So this Castile soap cleanser changed the microbiome in a sense that you see a lot more staphylococci and a lot of other microorganisms in their community structure at the end of the study. So what we're looking at now is trying to understand what the difference is in this community structure versus the community structures that we see here, to understand what the feedback of these microbiome communities might be to the skin once you perturb them with cleansers that change the pH and the barrier function of the skin. And so to summarize, I wanted to tell you that the skin microbiome is emerging, and it's an integral aspect of our skin barrier. And so we need to learn a lot more about how this aspect of our skin is functioning, but I think we're beginning to learn more and more that it's important for us to respect our microbiome, particularly in situations where the microbiome may be contributing to diseases like atopic dermatitis or even acne. And the skin microbiome that we develop in infancy is different from that of an adult, as I showed earlier, and this ecosystem continues to evolve. It even continues to evolve throughout the older years of life. So as your skin dries out, as you get older, your microbiome community continues to change. And the skin microbiome, the changes that occur after repeated use of various products that are not designed to respect the skin's barrier function, may impact long-term health. So these minor perturbations may add up to long-term issues in the long run. And an altered microbiome, as I mentioned, is seen in skin diseases. And we know there's a contribution of Staphylococcus aureus to atopic dermatitis. And so it may represent a new way to harness healthcare, or therapeutics rather, for this disease. And so limiting the distribution of these changes, too, by using the right kind of products that respect the skin barrier may help prevent the development of flares or keep the flares from happening as often. So thank you very much.
It's now my pleasure to introduce Professor Joseph Fowler, who is Professor of Clinical Dermatology, University of Louisville, and Professor in the Occupational Medicine Department, University of Kentucky. Professor Fowler has been a dermatologist for almost 30 years. He has an international reputation in irritant contact dermatitis, skin allergy, and rosacea. And he's now going to discuss the clinical implications of formulations we put onto the skin, and how all of this beautiful science translates into the products we put onto the skin and the science and art of formulation. Professor Fowler.
Thank you, Professor. So yes, let's talk about some things that we can do now. We as clinicians, we're treating our patients. We now know with this background some of the reasons why some of the options we've had in the past have been useful, and we're going to talk about what we might have in the future to come as well. We've seen and we know now in the last 10 or 15 years of research that patients with atopic dermatitis have barrier abnormalities due to poor NMF production, due to ceramide and lipid imbalance, which I'll talk about a bit, other genetic factors influencing the microbiome. As we know, atopics do not deal well with bacteria on the skin. The inherent innate immune system is usually defective in the atopic. What are we going to do about all that? Another thing I want to mention briefly, because I have a great interest in allergic contact dermatitis, is that recent work here in the UK and in Scandinavia has shown that, in fact, individuals with atopic dermatitis and filaggrin defects are at more risk for developing allergic contact dermatitis. Something we certainly have to keep in mind when we're treating our patients. We would obviously like to give them products, recommend products that are useful for them. We certainly want to not recommend products that would be perhaps allergenic to them. We know that moisturizers are the foundation of our treatment of atopics. We've known this without knowing perhaps why. We've known this for decades, maybe centuries. And most moisturizers, most basic moisturizers, can confer some level of barrier protection. We're going to talk a little bit about some of the newer agents and the newer components of moisturizers. You've already heard some, especially from Professor Cork, that I think enhances our ability to treat the individual with atopic dermatitis. And by corollary, those with irritant contact dermatitis, chronic hand eczema, that sort of thing, I think fits into these categories as well. So first we had simple occlusives. What I like to think of moisturizers as sort of a first, second, third generation process. Our first generation were simple occlusives such as petrolatum, mineral oil, and so forth. When I'm giving talks at meetings, it's not uncommon for someone afterward to say, oh, well, I always just recommend petrolatum for my patients. It's inexpensive. They do just fine with that, and I don't need anything else. Well, I think you've already seen that that is probably not the case, and we're going to talk some more about that as we go. I think there's no question that, especially in the atopic or the individual with, say, long-term irritant contact dermatitis, we'll see much better benefit by moving beyond those first-generation moisturizers. Temporarily, they work great. If you produce some irritant contact dermatitis on the skin and put some petrolatum on and measure transepidermal water loss in a few hours, sure, it's improved. Transepidermal water loss has gone down. The barrier is a little better by that measure. But then you wait another few hours, and that benefit is totally gone, totally lost. And so unless you're smearing that petrolatum-based moisturizer literally on the skin almost continuously, at least every few hours, that's not going to be a good option for your patient. Is that going to be a good option, that frequent moisturizing for your patient? I think not. You know, the products often are very greasy. Adherence, we'll talk about, is critically important. So while those simple moisturizers may work temporarily, in the long term, they certainly don't seem to be as effective as some of the better products that we have. So then the second generation moisturizers came along, and we added various humectants, maybe added some components such as glycerin, natural moisturizing factor components, perhaps not really understanding why these things were helpful, but clinically it seemed that some of these components helped. And that it was all great, and that reduced the need for maybe the greasy petrolatum-based moisturizers. But we were starting down that direction some years ago to get a more optimum moisturizer, one that helped to reduce the pH of the skin, as we've seen is so critically important, and helped to help the feel of the skin, the smoothness and roughness of the skin, help to improve that. But if we have the best moisturizer in the world, and if a patient doesn't use it, then it's not going to do any good. And so adherence or compliance is absolutely critical. I think we all understand that if we can increase the use of a good moisturizer, we will not only help the patient's skin do better, we will help them reduce the need for prescription topical therapies, which of course would reduce cost, may reduce potential adverse effects. There was a survey done here in the UK not long ago asking individuals about a colloidal oatmeal-based cream and a shower oil as part of their management of dry skin or eczema. You know, colloidal oatmeal has been around since the Egyptian times. I think anything that's been around that long, there must be something good about it. Now we understand the science behind it a lot more than was understood years ago, but certainly it's been a product around for a long, long time. And in this survey, subjects were asked to use the colloidal oatmeal-based cream and shower oil over the course of a couple of weeks. And at the end of this survey, the vast majority, over 90%, said, hey, this is really good, we like this, I would recommend this. This was better than what I was doing before. We'll talk more about a specific look at research into that as well in a minute. So let's talk about a few of the things, if we're recommending products for our patients, let's talk about a few of the things we ought to look for and ask about in a particular product. Maybe some things that should be good and some things that maybe we would try to avoid. Certainly, we've learned that natural moisturizing factor components would be nice to have in a moisturizer. It would be nice if we could enhance the inherent production of those, the endogenous production of those, as Professor Cork showed in his slide. It would also be nice if we could enhance the ceramide levels and perhaps the other lipids on the skin, cholesterol and fatty acids. That would be a nice thing to improve the skin barrier, it would seem. And it turns out that colloidal oatmeal contains a variety of lipids, contains naturally occurring components of natural moisturizing factor, as some polysaccharides, for instance, that endogenously could perhaps increase and improve our skin barrier defect in those individuals. Just clinically looking at a colloidal oatmeal containing lotion, a colloidal oatmeal containing skin relief lotion compared to vehicle is what we're seeing here. And you know, rarely do you see this type of a study. We often see vehicle-controlled studies with corticosteroids or calcineurin inhibitors or what have you. But how often do you see a study looking at one particular presumably active ingredient, in this case colloidal oatmeal, and comparing it with its vehicle? So the only difference in the dark bar and the lighter bar is the fact of the colloidal oatmeal component of that product. And what we can see here is a highly statistically significant increase in skin hydration as measured by corneometry after the several weeks, 3 or 4 weeks of use of the product. And while there was certainly increase in moisturization or skin hydration with the non-oatmeal containing product, it was obviously much greater with the oatmeal containing product. Clinically, as Michael said earlier, you don't see as much difference. It's a more crude measurement. But even clinically, while again there was improvement in scaling, dryness, and pruritus with the use of both products, there was greater improvement with the colloidal oatmeal product. And then, of course, when our patients are brought into the equation, if a patient won't use the product, what good does it do? In this situation, in that study, about 90%, 80 to 90% or so of patients preferred the colloidal oatmeal-containing product to the identical product without the colloidal oatmeal in many ways, as far as improving pruritus, improving skin dryness, making the skin feel softer, smoother, and obviously that would lead to greater adherence. Now, what about some of the things we should watch out for in our products that we're recommending to our patients? We know that fragrances and preservatives are major allergens. Certainly not everybody in the world is allergic to these things, but if you look statistically, for instance, in our data from the United States, from North America, we see that about 10% of the patients that we patch test are allergic to fragrance allergens, and about 10% are allergic to various preservative allergens. The formaldehyde-based preservatives such as quaternium-15 and others, and methyl isothiazolinone, or MI, are particularly problematic in that area. So if we're treating someone, especially chronically, with atopic dermatitis or long-term contact dermatitis, hand eczema, what have you, I would like to not use those components in our patient's treatment. Propylene glycol, we've probably all heard about as dermatologists. There's kind of a knee-jerk reaction to say that propylene glycol is probably not a good thing. But in fact, if it's in a low concentration, it's not bad. We may not always know what the concentration is in a product. Certainly high concentrations of propylene glycol can be very irritant and allergenic. Lanolin likewise can be allergenic, although some components of lanolin are not so much. And a variety of surfactants, of course, we have to be thoughtful about. You heard a lot about the harsh surfactants such as sodium lauryl sulfate earlier. The synthetic detergents or syndats such as cocamidopropylbutane are not nearly as irritating, but they're certainly more likely to be allergenic. Well, okay, so products have to have a preservative in most cases. If there's water, there must be a preservative to guarantee any sort of shelf life. What are the better ones? Parabens by far are much less allergenic and much less problematic than most of the other preservatives we have available to us. Unfortunately, and I think erroneously, parabens have been removed from a variety of products when in fact I think they are, from our dermatologic safety standpoint at least, some of the best. Phenoxyethanol is another one that can be used that rarely causes skin allergy or irritancy. Patients like products that smell good, of course, and again, for adherence, they don't like products that smell badly. Very obvious. But we know that many fragrances are allergenic. What about if we just used a simple skin conditioning agent that has a bit of a scent, like a lavender or a chamomile? Some patients with fragrance allergy may not tolerate those, but we think, and in fact studies are in progress now to try to understand that some of these skin conditioning agents that have a little bit of scent might indeed be okay for our patients, even our fragrance allergic patients. Natural moisturizing factor has been talked about quite a bit, and it's what holds water into the skin. It's in the cornea site. It's what helps hold water. I like to think of the skin barrier as water and lipid, and we've got to have the balance of those two. This is what's in natural moisturizing factor. We'll have a test on this later, and there'll be extra dessert outside if anybody gets these all right with the percentages. But you can see, as Michael mentioned earlier, PCA, for instance, is critical, and he showed how that could be enhanced with the proper moisturizing use. Filaggrin, because it's defective in most atopics and in other conditions, such as individuals with ichthyosis, the filaggrin defects in breakdown are critical to the atopic pore production of natural moisturizing factor. You've heard about that. We won't go into that further, but what about the lipid components? Cholesterol, ceramides, fatty acids, that's the mortar of the brick and mortar concept that we like, and this just tells us that the ceramides are important, more so perhaps, the quantity is greater. They're not more important than these others because the balance is very critical. So this is another area similar to the natural moisturizing factor slides we looked at earlier, with ceramide components of the skin. And on the left, what you're seeing is a bunch of healthy controls and looking at the total ceramide levels in some of the various subgroups. Here in the middle, we have atopic individuals, but in normal-looking skin. And here on the right, we have lesional skin of atopic dermatitis, so active disease over here on the right, and non-active disease here in the middle. Well, you can see that both of those groups with the atopic individuals had decreased ceramide levels, which puts that lipid component out of balance in their skin. Is it because of the higher pH they have? Is it because of an intrinsic genetic defect? There perhaps are a variety of reasons, but the point is that these folks who have poor ceramide production perhaps could be improved if we could enhance that ceramide production. Ceramides are long-chain lipid structures. There's a big long chain down here, and then the fatty acid component up here. So what if, we showed earlier, Michael showed a slide where PCA could be enhanced by the use of a proper moisturizer with a humectant. Well, let's look at the future. What if we could enhance endogenous production of some of these other components that are critically lacking in the atopic? This is a small study just presented recently at the World Congress of Dermatology in Vancouver less than a month ago, looking at a moisturizer, a unique gel matrix moisturizer. When applied to the skin, this gel matrix breaks apart, allows the water and the humectants in this matrix to enter the skin, and it contained emulsifiers that helped to mix in the lipids so that it was a cosmetically acceptable product, not a greasy feeling product, in other words, something that patients liked to use. Well, in vitro, this seems like a good idea. In vivo, what I think is particularly interesting is some of this first part here that we'll look at. When this moisturizer, this gel matrix moisturizer that did not contain ceramides itself, was compared with a ceramide-containing moisturizer. So in the yellow, a commonly used moisturizing lotion that did contain ceramides. And I should mention, by the way, that when we see the word ceramide on a label, the first question probably should be to the producer of that product is how much ceramide is really in there, because ceramides are quite expensive. And it may well be that there's a little bit of ceramide in some products, and maybe that's not enough to do much good. So in this case, this gel matrix moisturizer was compared to a ceramide-containing moisturizer. And what we're seeing here is that this gel matrix moisturizer turned on genes that synthesize ceramides quite effectively, much, much more than the ceramide-containing moisturizer. And we also then saw that treatment with the gel matrix moisturizer over the course of about 4 weeks resulted in a significant increase in ceramide production in the skin, as you can see compared to baseline, much greater ceramide level. And while treatment with the ceramide-containing moisturizer improved a little bit as well, it didn't give you as much improvement as this gel matrix moisturizer, telling us then that the moisturizer itself was able to inherently upregulate production of ceramide in the skin, exactly the sort of thing that we need to do, I think, in the future for enhancing the skin barrier. So I think it's going to be critical in closing to say that it looks like we're on the frontier. We've developed some new moisturizers, some new components. We have a much better understanding now than we did 20 years ago of what we need to do to try to enhance the skin barrier in the atopic. We're on the frontier, I think, of getting products now that we can recommend with confidence to our patients that we actually are going to enhance that skin barrier rather than impair the skin barrier further than it is already. And I think we're going to do that in the near future. Thank you all very much.
Joseph, thank you very much. That was an eloquent illustration of the effect of the next generation moisturizers, emollients, how they can actually induce expression of ingredients in the skin. And it reminds me of the last generation emollients. I remember David Page wrote his MD thesis on ceramides in different formulations, and he actually analysed a cosmetic moisturiser many years ago and found one molecule of ceramide in the entire bottle of moisturiser. It's good to see the science has moved on a long way since then.